prostate. It's at the base of the bladder. When the urethra runs through the prostate gland, it's called the prostatic urethra. Then the urethra goes through this band of muscle right here. It's a pretty thin band of skeletal muscle, but it's still a band of muscle. That band of muscle is called the urogenital diaphragm. So it's just skeletal muscle. But when the urethra runs through that part, which is not a very long part of the urethra, but when it runs through there, it's called the membranous urethra. And then the longest part of the urethra goes through the penis. So it would start here, and then it would come all the way through the penis. That part of the urethra is called the spongy urethra or the penile urethra. Okay? So it's all the urethra. It's kind of like a road as it changes names depending on what it crosses with. It's the same basic thing. It's still just one tube. Okay. All right, so we're going to start where sperm are produced and then follow how the sperm mix with secretions upon ejaculation and how all of that, the secretions plus the sperm together equals semen, how that exits the body. Okay, and again, we'll get into all this stuff in more detail throughout the rest of the lecture. Okay, so the testes, the gonads, right here, they are housed outside of the abdominopelvic cavity. They're housed within this sac of skin called the scrotum. And we're gonna have to talk about why would you put your reproductive organs that make your gametes for propagation of the species, why you would put that outside of the body, okay? That's a little risky to do that, right? But we'll talk about why that's the case. So, sperm are produced in the testes, literally in the wall of the testes, and we'll look at the details of that later. Sperm are produced here. The whole process of making a sperm takes about 9 weeks. And so sperm are continuously being produced from puberty throughout the life of the male, assuming that hormone levels stay constant. We'll look at all that stuff later. Very different from females. Okay, very, very different from females in that regard. But from puberty on, sperm are always being produced, and it takes about 9 weeks to make a spermatozoa. They're going to leave the testes, and then they next go to this structure here called the epididymis. The epididymis. The sperm stay in the epididymis to continue maturing. If you were to pluck a sperm out of the testes, it could not fertilize an egg at that point. It still has to continue maturing, and it has to mix with all these secretions that are released upon ejaculation. Okay, so it needs all of that in order to fertilize an egg. So sperm are produced in the testes. They next make their way to the epididymis. They're stored in the epididymis, continue maturing. If they're damaged, they'll get broken down and their base components just reabsorb back into the body. Okay, that happens in the epididymis. If ejaculation occurs, then the sperm will be propelled out of the epididymis into this tube, this passageway, this tube that goes from outside the abdominopelvic cavity to into the abdominopelvic cavity, comes around the anterior side of the bladder, the superior side of the bladder, and the posterior side of the bladder. And that tube is called the ductus deferens or the vas deferens. Somewhere on there. Here we go. Ductus deferens or vas deferens. Okay. So sperm are made in the testes, they go to the epididymis, they continue maturing at the epididymis, they're stored in the epididymis. If they're damaged, they'll get broken down and basically components reabsorbed at the epididymis. They'll stay there for about 2 weeks or so, unless ejaculation happens beforehand. If it doesn't happen by about 2 weeks, then again, they'll get broken down and the base components reabsorbed back into the body. If ejaculation occurs, then the sperm will be propelled out of the epididymis through the ductus deferens. Then the backside of the ductus deferens gets enlarged, okay, it just gets bigger. That's called the ampulla of the ductus deferens, just where it gets bigger. And the sperm that are in the ductus deferens, in the ampulla of the ductus deferens, where they go next is into this short little tube right here that runs through part of the prostate gland. And that little tube right there is called the ejaculatory duct. Now, this is where we're gonna get our first major set of secretions mixing with the sperm. Upon ejaculation, not only are the sperm propelled through the ductus deferens and then into the ejaculatory duct, but we also get 3 sets of glands contracting and releasing their secretions. The first set comes from this gland right here called the seminal vesicles or the seminal glands. So secretions from the seminal glands and sperm that's traveling through the ductus deferens, they mix together here in the ejaculatory duct. Then from the ejaculatory duct, we go into the prostatic urethra. Okay, prostatic urethra, and then we get our next set of secretions coming from the prostate gland itself. So we've got 2 sets of secretions so far, from the seminal vesicles and from the prostate gland, mixing with sperm that were made in the testes that have been sitting in the epididymis maturing. Then, as we go through the prostatic urethra, we then go through the membranous urethra, and then into the first part of the spongy or the penile urethra, and this is where we get our 3rd set of secretions from this little gland right here. This little gland right here is on the posterior side of this urogenital diaphragm, and on the models upstairs, most of them you can't see that little gland right there, but you should know that it's there, okay? So on your practical, I might put a tag right there and say, you can't see it on this model, but there's a gland here, what's it called, okay, or something like that. This last gland is called the bulbourethral gland, okay? And again, it's gonna contract and release its secretions, and at this point, all those secretions and the sperm together make up semen. That semen will continue traveling through the rest of the spongy or the penile urethra, and out the body it goes, okay? So one more time. Testes, sperm go to the epididymis. Upon ejaculation, epididymis contracts, propelling that sperm through the ductus deferens. From the ductus deferens into the short ejaculatory duct that runs through the prostate gland. We pick up our first set of secretions from the seminal vesicles. Then those secretions from the seminal vesicles and the sperm travel next to the prostatic urethra that again runs through the prostate gland. We pick up our second set of secretions from the prostate gland. Then those 2 sets of secretions and the sperm next travel through the membranous urethra and then into the top part of the spongy or the penile urethra, picking up our 3rd set of secretions from the bulbourethral gland. At this point, we've got 3 major sets of secretions and sperm. Collectively, that's called semen. It will finish traveling through the spongy or the penile urethra and out the body it goes. Okay. We'll go back and talk some more about that stuff. But you should be able to identify those structures on your model upstairs, and for your lecture exam, know the order that this all travels through. Okay, so let's talk about some of these structures. So the scrotum, we said that this is essentially a sac of skin and connective tissue that houses the testes. And we want to say why? Why are the testes outside of the abdominal pelvic cavity? And the reason they're outside of the abdominal pelvic cavity is because sperm are really particular cells. They're really small cells, and we'll talk about why they're so small and how they get to be so small. And they're really particular about the conditions in which they develop, okay? And then once you have a mature sperm, they're still particular about the conditions in which they are residing. In order to produce sperm, the body cavity temperature is too hot, okay? It just does not provide enough viable sperm if you do it in the body, inside the body cavity. So that's why they're made outside of the abdominal pelvic cavity in the testes that are outside. That allows the testes to be about 1 to 3 degrees Celsius cooler than body temperature, okay? Not Fahrenheit, but Celsius, okay? Cooler than body temperature. But this also then means that we have to have a way of regulating the temperature of the testes because we can't control the outside environment. If the outside environment gets too cold and the testes get too cold, that's not so good for sperm production either. If it's hot outside, outside and the testes start to get too hot, that's not so great for sperm production either. So what we have are two sets of muscles that can help to either warm up the testes or cool down the testes. These two sets of muscles, one set is called the dartos muscles. These are smooth muscles that are in the dermis of the scrotal skin, basically a layer of the skin of the scrotum. When these muscles contract, remember smooth muscle does a twisting motion, not a shortening motion. When these smooth muscle cells contract, they twist, and what that does is it causes the scrotal skin to wrinkle and kind of tighten everything up. That decreases surface area, and as you bring everything closer together, it helps to warm the testes. Okay, so you're basically bringing all the skin and connective tissue closer together, and that helps to raise the temperature. At the same time that's happening, you've got another set of muscles called the cremaster muscles that are skeletal muscles. When these contract, it elevates the testes so that they're closer to the body. So if the testes are too cold, reflexes will kick in that cause these two sets of muscles to contract. The dartos will cause wrinkling or contraction of that scrotal skin. The cremaster will elevate the testes, and all that increases the temperature of the testes. If the testes are too warm, then these sets of muscles are going to relax more. And it's a matter of contracting more or relaxing more. It's rarely that they're completely one or the other. It's usually some variation in between. So if the testes are very warm, then those sets of muscles are going to relax more. As the scrotal skin is not wrinkling so much, as that muscle relaxes more, it allows the scrotal skin to flatten out and surface area gets bigger and heat can be dissipated. The cremaster muscles will allow the testes to depress further away from the body. Again, that helps to dissipate heat. And if it's further away from the body, it's not picking up the warmth of the body as much. Okay, so that's how we're gonna do that. Okay, so this right here is looking at a coronal cut like this. Okay. This is a nice image because what you can now see is you can see the scrotum here, and then inside you can see testes. Over here, the connective tissue has been pulled off so we can kind of look inside. And what we see is, we see this cord right here. So this is not a tube or a passageway, but this is a cord containing various structures. And this cord is called the spermatic cord. And you guys are gonna look at a cross-section of a slide of a spermatic cord when we're upstairs. And what you're gonna see in the spermatic cord is a lot of very small arteries, a lot of very small veins, cross-section of some nerves, and then also a cross-section of a ductus deferens. Remember, that's that tube that takes the sperm from the epididymis into the ejaculatory duct. So in the spermatic cord, there would be one ductus deferens in here, a bunch of small nerves, a bunch of small arteries, and a bunch of small veins in that spermatic cord. What else do you want to say about this? And you can see the same in the other spermatic cord. The nerves and the blood vessels are just supplying, so the blood vessels are supplying nutrients and taking away waste from all the tissue in the epididymis, the scrotum itself, and the testes, and then the nerves are supplying neuroinnervation to the muscle down there. I guess while we have this slide up, let's say a couple things about vasectomy since we have this slide up. Vasectomies are on a different slide, but while this is up, this is a nice time to talk about it. You guys probably aren't at that point in your life where you're really thinking too much about children, but if and when you get to that point, or thinking about maybe after you've had, well, whatever, okay? I'm not gonna counsel you on having children or not, but if you're looking at things like tubal ligations for females versus vasectomy for males. For females, to do a tubal ligation, that is major surgery, okay? You have to put the female completely under with anesthesia. You have to cut into the abdominal pelvic cavity, you have to cut through all the muscles, you have to open that up, you go in there, you find the 2 fallopian tubes, you cut them, and then you ligate them, okay? It's major surgery, recovery time takes a while, it's major surgery. For males, a vasectomy is not major surgery, okay? They don't have to be completely under, you can do this in a doctor's office. Recovery time is like that, relatively speaking. What you do for vasectomy is you can go in here outside of the abdominal pelvic cavity, you can come into the spermatic cord. You can do that for each of them. You cut into the spermatic cord, obviously there would be a local given, you cut into the spermatic cord, you find the ductus deferens in each of these, you cut the ductus deferens and you ligate it, okay? Nothing else is altered. Sperm are still being produced, hormones are not affected whatsoever, ejaculation will still occur, you still produce semen, it's just that the semen lacks sperm. Everything else is the same. Sperm will still be produced, sperm will still go to the epididymis, but upon ejaculation, as the sperm start traveling through the ductus deferens, they can only get so far because it's cut and tied. Secretions are still coming from the seminal vesicles, the prostate gland, the bulbourethral gland. It's just sperm won't be in those secretions, okay? The sperm will make their way up to the ductus deferens as far as they can, and then they basically will fall back down. They eventually will get broken down and the base components reabsorbed. Just like what would happen if the sperm were sitting in the epididymis and ejaculation didn't occur for a while. Same basic thing. Proteins will get broken down into amino acids, they'll be put back into the bloodstream to be used by other cells. Carbohydrates will get broken down into glucose, glucose will get put back in the bloodstream to be used by other cells. Same basic thing. The only thing you have to worry about with a vasectomy is just making sure that you go back into the follow-up appointments where they'll test the semen to make sure that there are no sperm in there. Sometimes what can happen is the 2 parts of the ductus deferens can fuse back together. They can essentially grow back together. And then you don't have a cut and a tie anymore because it's come back together. But that's a quick and easy check. You just look to see, are there sperm in the semen? If there are, well then something didn't work right. Sometimes, instead of having 1 ductus deferens per spermatic cord, sometimes you get 2 ductus deferens per spermatic cord. But again, all those things are easy checks. And for the most part, vasectomy is reversible. So if they go to reverse a vasectomy, they just take the 2 ends that have been cut and tied and they just suture them back together. And as long as there's not a lot of scar tissue when they suture that back together, as long as there's not a lot of scar tissue and the sperm can get through where it's been sutured back together, then everything's back to normal. Okay? So much, much, much safer, easier, cheaper, on and on and on. This is vasectomy versus a tubal lift. Okay, testes. Let's take a look at the testes. We're going to look at the testes right now, talk about a few other structures, and then come back to the testes to look in detail about how sperm are made. Okay, so we're going to loop back around. But let's do a first run through here. So what we're going to do is we're going to look at the next slide. When we look at the next slide, we're going to see that the testes are subdivided into what are called lobules. Okay? And in between these lobules is dense connective tissue, which is just called a septa. It's just fibrous partitions, dense connective tissue, lots of collagen fibers, essentially just roping off one lobule from another lobule. In these lobules are very slender, really tightly coiled tubules, kind of like we saw with our renal tubule, okay? But these are more slender, and they're even more tightly coiled than our renal tubules were. These tubules are called seminiferous tubules, okay? And it's in the wall of these seminiferous tubules, that's where sperm are produced. And again, we'll look at that in more detail in a minute. If you take a testes and you pull out the seminiferous tubule from each testes and put those tubules end to end, straighten them out, put them end to end, we have about a half a mile tubule per testy, which really is amazing when you think about how big a testy is. And then half a mile of tubule coiled up inside of that. It's just amazing. But in the wall of the tubule is where you make sperm. And then around those seminiferous tubules, you have other cells around the tubules. And some of those cells that are around the tubules are called interstitial cells because they're in the interstitial space around the tubule. They're also called cells of Leydig because that's the person that discovered them many, many moons ago. But it's these cells, these interstitial cells, that produce the androgens, the testosterones. And so when we talk about hormones, we'll come back and talk about our gonadotropins, FSH and LH, and talk now specifically which one of those targets which of these groups of cells. One's going to target the seminiferous tubule, the other's going to target the interstitial cells, and we'll clarify that later. Now, once we make the sperm, again, they're made in the wall of the seminiferous tubule. Once they are done, once we produce spermatozoa, they will detach from the wall. So let's say we're in the lumen of a seminiferous tubule, okay? So let's say we are sperm, okay, in the lumen of this seminiferous tubule, okay? We originally were made in the wall, okay, in this wall. So where we would have started the process would have been on the far part of the wall, so like this part out here. And as the cells divided, the daughter cells from undergoing mitosis and meiosis, they kept getting from the outside here of the wall, they kept getting closer and closer and closer to the lumen. Until eventually, what we'll end up with is the head of the sperm will still be stuck in the wall, and the tail will be hanging out here in the lumen. And the last part of making a sperm is it detaches from the wall, and now it starts swimming, okay? Swimming through this lumen. It will travel through the lumen of the seminiferous tubule and start making its way to the tubules that are in the epididymis. Now, we are not going to learn the names of all these tubules between the seminiferous tubules and the tubules in the epididymis. We're not going to learn the names of all those. And here's some of the names that we're not going to worry about. Okay? So we're just going to say that sperm are made in the wall of the seminiferous tubule. We'll look at that process in detail very shortly. They detach from the wall of the seminiferous tubule, and then they start swimming through a series of tubes, passageways, until they get to the epididymis, which is also a series of tubules. Now, when we're upstairs in the lab, we are going to look at a cross-section of the spermatic cord, which you're going to look for cross-sections of arteries, veins, nerves, and the ductus deferens, and I'll go over that with you when we get up there. You're also going to look at a cross-section of the testes, and you're going to look at a cross-section of the epididymis. Okay? And the testes and the epididymis both are going to be cross-sections of a bunch of tubules. And we'll talk about later how you can tell the seminiferous tubules of the testes versus tubules in the epididymis. And we'll get to that later, but you can definitely tell the difference. Okay, so this is what we were just looking at. So cross-section this, we can see these are lobules right here. In the lobule, you'll have seminiferous tubules, that's the yellow thing. The white part that separates one lobule from another, that's called septa. It's just dense connective tissue. And what they don't show here, that around all these seminiferous tubules, so it would be in this white space here. All around here would be interstitial cells. Okay, so they would be around the tubule. Okay, so sperm are made in the wall of these tubules. They detach from the wall, they swim through the tubules, they swim through these tubules, and eventually they make their way into these tubules here in the epididymis. And then eventually upon ejaculation, they'll leave the epididymis and go into the ductus deferens. Okay, let's say a couple things about the penis. Penis obviously is the copulatory organ. Its purpose is to deliver sperm into the female reproductive tract. Now, let's see, let's talk about a couple key features here. So this right here is looking at a coronal cut. Here's the bladder. We're looking at the posterior view, okay? We're looking at the bladder. Here's our ureters. I know this is posterior because I can see my seminal vesicles. Seminal vesicles are on the posterior side. At the base of the bladder would be the prostate gland. We've got our ductus deferens coming in. The ductus deferens expand into the ampulla, and the secretions from the seminal vesicle and the sperm in the ampulla of the ductus deferens would come into the ejaculatory duct that runs through the prostate gland and then into the prostatic urethra. We can see that prostatic urethra then goes into the membranous urethra as it runs through this band of urogenital diaphragm, and then into the spongy or the penile urethra until it exits the body. When we look at the tissue that makes up the bulk of the penis, you essentially have two different groupings of what's called erectile tissue. Erectile tissue is this kind of spongy network, kind of like last quarter when you talked about cancellous bone, where your bone marrow was. It kind of had this spongy or lacy appearance to it with a bunch of like cavities within it. That's kind of what this looks like. So it's a spongy network of connective tissue and smooth muscle. Okay. And all within it, you've got these just like kind of pockets of space, just empty space. And what can pool in there is blood. Okay, so that's what makes it erectile tissue, is blood can essentially pool into these little spaces called vascular space. So the way the penis becomes erect is blood pools into this vascular space, causes the penis to become erect, and then as that blood drains away from that vascular space, the penis becomes flaccid again. Now, when you look at this erectile tissue, if you were to look at it under the microscope, which we're actually not going to do, in terms of how the tissue is organized, it essentially looks just like the clitoris in females. It's both erectile tissue. It engorges with blood. There's lots of nerve endings associated with both. Okay. When we look at the male erectile tissue, we've got two groupings of it. One is called the corpus spongiosum, and this you'll need to know for lab. Okay? So for the corpus spongiosum, and then you have what's called the corpus cavernosa, or sometimes instead of corpus, you'll see that called the corpora. Corpora cavernosa. And that's because the corpus cavernosa is paired. It's a set of columns. So corpora just is referring to plural. Your corpus spongiosum, you only see that called corpus because there's only one of them. Okay, so corpus cavernosa, there's two of them, so you might see it called corpora cavernosa. I'm not going to grow you on singular versus plural. So where do you find these and what's their purpose? So corpus spongiosum, this is going to be a circular column of erectile tissue that goes right around the urethra. Okay, so if we could kind of make a circle, it would go around the urethra like this. Okay, so we come out and go around. If we cross-section this and look, like if we were standing on top and looking down, here's the urethra, and going in a circle around it, that's this corpus spongiosum. It's not a lot in terms of erectile tissue, there's not tons of it there, but its purpose is to make sure that the urethra doesn't collapse when the penis engorges, when the other erectile tissue engorges with blood and the penis becomes erect. It makes sure that makes sure that the urethra stays open so that semen can run through it. So that's the main job of the corpus spongiosum. Okay? Its main job is to make sure the urethra doesn't collapse on itself. Then you have these paired columns that sit dorsally, okay? And that's these two right here. They're the majority of the erectile tissue that make up the penis. So over here, it would be these columns here, and they would sit dorsally, okay? That would be these guys. Okay. And when they, their main function is to engorge with blood and cause the erection. Now, the corpus spongiosum also is engorging with blood. And though it does contribute to the erection, its main job is to make sure the urethra doesn't collapse. Okay, all right. Okay, so then let's go to the epididymis and say a couple things again about the epididymis. So I said you're going to look at a cross-section of the epididymis and you're gonna see tubules. The way you're gonna tell the epididymis from the testes is when you look at the testes and you look at a cross-section of the seminiferous tubules in the testes, what you're gonna see in the wall of that seminiferous tubule is a whole bunch of cells that are not in defined layers, that are in the process of cell division. So you've got cells doing mitosis, you've got cells doing meiosis, because they're ultimately making sperm cells. If you look in the wall of the epididymis, you don't have that. If you look in the wall of the epididymis, the tubules in the epididymis, what you're gonna see is a layer of epithelia, right, lining a lumen. And the epithelia that you're gonna see here in the epididymis is pseudostratified, columnar epithelia. And it's going to be very distinctive. You're going to see a single layer of cells. You'll be able to see their nucleus very clearly defined. Whereas in the testes, as cells are undergoing mitosis and meiosis, you can't see their nucleus anymore because there is no nucleus. The nuclear envelope breaks down. What you can see in the cells undergoing mitosis and meiosis in the testes is you can see chromosomes condensing. That's what we'll be able to see. Here, we'll be able to see a very clearly defined layer of epithelial cells. And on the apical surface, we will have very long microvilli on these cells. So on the apical surface, the surface that faces the lumen, so that's the lumen side, we will see very long microvilli here. And unfortunately, long microvilli has the unfortunate name of stereocilia. I dislike that name because when I see stereocilia, the first thing that comes to my mind is cilia. And these are not cilia, they are microvilli. And they are not the same thing. Cilia does what we saw in the reproductive tract, move in a coordinated sweeping motion. Whereas microvilli is there only to increase surface area, right? So these are long microvilli with an unfortunate name. So why do we have microvilli here? We have microvilli here because here in the epididymis, we can recycle damaged sperm. So as we recycle these damaged sperm, all those base components like amino acids and glucose and fatty acids are getting absorbed back into the body. And having more surface area means that we can absorb them better, faster, more efficiently. If we've got more places to absorb, we will absorb better. We're also gonna see that we do get a little bit of secretion happening here at the epididymis as well. We get a little bit of fluid that is being secreted to mix with that sperm as well. And again, that increased surface area just helps all that absorption and secretion to just happen better, faster, more efficiently. Lastly, the epididymis is going to help to store and protect this sperm until it's ready to be ejaculated. Once it gets ejaculated, it can then mix with fluids. And once it mixes with fluids from the seminal glands, the prostate, the bulb urethral, then it will be able to fertilize an egg. But it can't fertilize an egg if you just pluck a sperm from the testes or from the epididymis. It's not quite ready yet. Now, when ejaculation occurs, there is smooth muscle in the wall of the epididymis in the wall of these tubules that will contract, and that helps to propel the sperm forward. So the sperm, though they have flagella, they cannot swim the entire way through the male reproductive tract and the female reproductive tract. It's just too far. They cannot swim the whole way. Their flagella helps them to move, but you also need contraction of smooth muscle. You've got to set up peristaltic contractions. Peristaltic contractions are the contractions that you see moving food through your GI tract, moving feces through the colon, moving cells through the reproductive tract. So peristaltic contractions is where you have smooth muscle contracting in a particular order that helps to propel things forward. So upon ejaculation, we're going to see that that is a sympathetic reflex. So ejaculation, and you'll see this on another slide. Ejaculation is under a sympathetic, so part of your autonomic nervous system, reflex. And that sympathetic reflex is going to cause smooth muscle in the wall of epididymis, in the wall of the ductus deferens, in the wall of the ejaculatory duct, in the wall of the urethra. Causes all of that smooth muscle in the wall of these tracts to contract, causing peristalsis, which again is just forward movement. Forward movement of sperm / semen. The other thing that the sympathetic reflex will do is it's going to cause glands to contract, releasing their secretions. And the three glands that we said are going to contract, releasing their secretions, are the seminal vesicles, the prostate gland, and the bulb urethral gland. I left out an O somewhere in there. Bulb And then lastly, 3. Remember we said that semen and urine can't go through the urethra at the same time. Part of this reflex will cause a sphincter, a bladder sphincter to contract, and that will prevent. This is also smooth muscle. That will prevent urine from exiting the bladder. We don't want we don't want urine and semen going through the urethra at the same time. Okay? There's multiple reasons we don't want that. And we'll talk about why the urine would likely destroy the sperm. We'll talk about that in a few minutes. So, upon that reflex, all kinds of things are going to be happening in the male reproductive tract that's going to start propelling this sperm and these secretions forward. So, as we leave the epididymis, we then will go into the ejaculatory duct, which again is pretty short. From the ejaculatory duct that runs through the prostate gland, we next will go into the, let me back up. We leave the epididymis and we go into the ductus deferens, From the ductus deferens, we then go into the urethra, from the ductus deferens, From ductus deferens, we then go into the ejaculatory duct, then the ejaculatory duct into the urethra. Sorry about that. And all of those have a nice thick layer of smooth muscle. In particular, the ductus deferens has a particularly nice thick layer of smooth muscle that we'll see under the microscope when we get upstairs. So this is just looking at the peristaltic contractions like we wrote up there. This is talking about vasectomy. And again, it's the ductus deferens that's in the spermatic cord that's outside of the abdominal pelvic cavity that gets cut for vasectomy. Okay, so then again, the urethra, we've got 3 different parts of the urethra. Urine and semen go through at different times. We don't want them going through at the same time. One key thing that we know about that we would want to avoid is the fact that urine tends to have a slightly acidic pH. Remember that we said that usually what we see at the DCT is that hydrogen ions get secreted. Hydrogen ions are getting secreted into the filtrate. That means they end up in the urine. Hydrogen ions being in the urine cause the urine's pH to drop. Urine pH tends to be around a pH of 6, can get down to a pH of 4 point something. Sperm are very particular. They don't like it too hot, and they don't like a low pH. Even a pH of 6, they don't like it. And that can cause them to become non-functional. So, of many reasons why we want urine and semen going through the urethra at the same time, one very key reason is the acidity of the urine would essentially cause the sperm to be non-functional. Okay, so let's say a few things about the secretions that come from these glands and what these secretions are there for. Like, what's the point of these secretions? We're going to talk about a couple different chemicals that you find in these secretions. And on your final, I will ask you something about this. Okay? It'll either be a multiple choice, a matching, or I might even say semen consists of sperm and secretions. Tell me 3 distinct compounds or chemicals that are in these secretions and what their function is. Okay, something like that. There'll be something about this on the test. So we want to then look and say, what kinds of secretions, what kinds of chemicals are in these secretions, and what are they doing for us? What's the point of those being here? Now remember, we're talking about these glands: seminal glands, the prostate gland, the bulbourethral glands. You do not have to say, this particular chemical is coming from the seminal glands, and this particular chemical comes from the prostate. You don't have to line up particular chemicals from the various glands. What I want you to know is those are the 3 major glands. They release secretions. I want you to know I want you to know that, and then I want you to be able to tell me a few very specific things that are occurring because of chemicals in these secretions. So first of all, there are chemicals. We're not going to name the specific chemicals in this example, but there are chemicals in these secretions that cause the sperm to become activated so that they can actually fertilize an egg. And we said if you just pluck a sperm from the testes or even the epididymis, it can't fertilize an egg yet because it's not quite mature enough. Part of that maturation process is to be in the presence of certain chemicals that come from these secretions that cause that sperm to become active so it can actually fertilize an egg. There are nutrients that are in these secretions that the sperm will utilize in order to make ATP. Now, what the sperm can't do is take some glucose with it. We can't store extra glucose as glycogen and put in that sperm cell because it would start to make the sperm cell too heavy. We're going to see that one of the big things that we have to do in making a mature sperm is make it as light as possible because it's got to go a long distance. Okay, it's gotta go through the whole male reproductive tract and through the female reproductive tract. And the female reproductive tract has a lot of dangerous barriers that the sperm have to get through. The female reproductive tract, for the most part, sees the sperm as a foreign invader. It tries to kill it, except for right around ovulation. It is a little bit more conducive to allowing the sperm to get through. But otherwise, female reproductive tract is essentially fighting off that sperm. So, in terms of nutrients, the sperm can't carry glucose with it, and even if it could, the sperm don't use glucose. Again, they're particular in that regard. Most cells like to use glucose to make ATP, not sperm cells. Sperm cells use fructose instead, fructose. Fructose is also a 6-carbon sugar, like glucose, but structurally it's a little bit different, okay? Fructose is what you find in the semen, and then the sperm can transport that glucose internally. The sperm have mitochondria. You can then take that take that fructose, and with the mitochondria, make ATP. And then the ATP is used to power that flagella, power the tail. The sperm aren't worrying about repairing its plasma membrane. The sperm aren't worried about making proteins along the way. The sperm aren't worried about doing anything except powering that flagella and trying to get to a I'm in quotes, trying to get to a fallopian tube. All right, so on your test you could say fructose is used as a nutrient to make ATP to power the flagella, okay, or something along those lines. Then, I had said that the sperm can't swim the whole way. They can't power their flagella and get through the whole male reproductive tract and the whole female reproductive tract. They just can't do it. So I said instead, what they need is peristaltic contractions to help, okay? They gotta have these peristaltic contractions with the flagella to get through the male and female reproductive tract. Now, to get peristaltic contractions in the male reproductive tract, that's easy. That's a reflex. That's under sympathetic innervation. Easy. But now, if the female is not having an orgasm, there will be no peristaltic contractions in her female reproductive tract. If she's having an orgasm, then she will have peristaltic contractions in her female reproductive tract, because smooth muscle in her tract will also contract and set up peristaltic contractions. But if not, then there's no peristaltic contractions in her tract. So what has evolved to get around that is, in the semen, there are chemicals called prostaglandins in there. And what prostaglandins do is they stimulate smooth muscle and cause contractions. So even if the female's not having an orgasm, the prostaglandins in the male's semen can cause her smooth muscle of her tract to contract and set up peristaltic contractions, so that the sperm is more likely to make it to that fallopian tube. Okay. So again, on your test, you could say prostaglandins are in these secretions. Their job is to cause smooth muscle contraction, or you could say peristaltic contractions of the female reproductive tract. Then, I said that sperm don't like a low pH, okay? Now, there's 2 places that sperm are potentially going to encounter a low pH of around 6-ish. One is, there might be residual urine in the urethra when ejaculation occurs. So if a male has urinated and then ejaculates within a certain period of time afterwards, there will be a little bit of residual urine in that urethra. And more critically, for what we're thinking about, is there will be hydrogen ions in that urethra, and that can cause the pH to drop. Also, when the semen enter into the vagina has an environment where there are natural flora there. Single-celled organisms, bacteria that are supposed to be there. They are endogenous. They are doing metabolic activities. And as part of those metabolic activities, just like our cells do metabolic activities, they produce acids. Those single-celled organisms, those bacteria that are there in the female vagina, that should be there in the female vagina, are setting up a slightly acidic environment. And that's beneficial for a couple reasons. It's beneficial because it keeps single-celled organisms and pathogens that we don't want there from taking root and residing there. So other bacteria, fungus, yeast, it helps to fight those off because they don't like that in acidic environment. But that acidic environment is not conducive to sperm. They don't like it. It can kill the sperm. So what has evolved is there are now buffers in the semen. The buffers in the semen help to buffer the male's urethral environment if it is slightly acidic, and it also buffers a potentially, a very likely pH of around 6, slightly acidic environment in the vagina. Now, sometimes this poses a problem for females, right? Sometimes having that acidic environment get buffered too much can then make a very ripe environment for other organisms to take root, like other bacteria, like yeast, like fungus. So that's kind of a mixed sword right there, right? Mixed bag. Okay, so you could talk about buffers, you could talk about prostaglandins, you could talk about fructose, and you could just say chemicals that activate spermatozoa, and not actually name those chemicals since we're not naming them. And again, if I were to say, where do these chemicals, where do these secretions come from? You just say collectively, they're coming from places like the seminal glands, the prostate gland, the bulbourethral gland. Okay, say a few more things about semen and some other chemicals and just other things that you find in semen. So when you look at volume, okay, the average ejaculate is not that large volume-wise, about 2 to 5 mils. It's really not all that much. But if you look at the number of spermatozoa that are in there, it's quite impressive, right? So in an average ejaculate, you have somewhere in the order of 20 to 100 million sperm per mil of ejaculate. So let's just take the high end of all this. Let's take 100 million per mil. If we have a 5 mil ejaculate, that's 500 million sperm, right? 500 million in that ejaculate. And so we should be thinking, wow, that's a lot. And that means that the testes have to make a lot of sperm constantly. And why? Why would they do that? Why would the body have evolved to put all those resources into making all these cells when really we just need one cell, one sperm to fertilize an egg? And the answer is, yeah, we don't really need just one to fertilize an egg, okay? So let's kind of clarify that first of all. So first of all, a lot of these, most of them are gonna die, okay? They're just not gonna make it. So think about what they have to, where they have to go. They have to enter the vagina, they have to get past the vagina. The vagina is slightly acidic. There are bacteria that are in the vagina. There are immune cells in the vagina. Sperm have to get through all that, okay? Then the sperm have to get past the cervix. The cervix is at the base or the neck of the uterus. And typically, there's a thick layer of mucus at the cervix that the sperm have to get through. And most of the time, they get trapped in that thick layer of mucus and they don't get past the cervix into the uterus. And again, that's a protective mechanism. When that cervix gets thin, when the mucus in the cervix gets thin, is right around ovulation. So I think it's like 12 to 24 hours, something like that, prior to ovulation, that mucus will start to get thinner so that the sperm can get past there easier. Birth control pills make that mucus thinner also. But normally it's pretty thick. And then let's say they do get past all that, and now they're in the uterus. Think about a tiny little sperm cell relative to a big hollow organ, a uterus. That's like a tiny little pebble in an ocean, right? It's just the size difference is huge. And then think about that uterus. If this is the uterus, right, and here's the cervix. Okay, so let's say the sperm, they get past the cervix, and now they're in the uterus here. There is going to be a tiny little hole here, and a tiny little opening here, I should say hole, opening, that leads into this fallopian tube, or this fallopian tube. Okay. So the sperm have to make it into one of those little openings, and then maybe ovulation is occurring at this point in time, and whether it's this ovary that ovulates or this ovary that ovulates an egg into their respective fallopian tubes, that's a completely random process. So you need enough sperm to get past the vagina, past the cervix, into the uterus, into each of those fallopian tubes in case there's an ovulated oocyte in one of those fallopian tubes. So you start with a large number because only a small number will actually make it to one of those fallopian tubes. And then let's say we do have sperm that make it to a fallopian tube where there's an ovulated egg. If you look at that oocyte, okay, so here's our egg, we'll just call it an egg for right now. If you look at that egg, there are barriers around that egg that must be penetrated. So what you need when you encounter an ovulated egg is you need anywhere from 10 to 100 sperm to start breaking down that barrier. Let's say that we do get a passageway formed by breaking down those barriers. We need about 10 to 100 sperm to do that, so that one sperm that still has all of its parts intact can come in here and fertilize that egg. Okay? So these sperm that are helping to break down the barrier, what they are doing is they are releasing enzymes from a part in the tip of the sperm's head, which is called an acrosome. We're going to look at that in a minute. Acrosome. So if you look at a sperm cell, we're going to see that there's different parts. You have what's called the head, the mid-piece, and then the tail. And at the head here, at the very tip of it, is a cap or a helmet that contains digestive enzymes. And that has to open and release these digestive enzymes to break down that barrier, so that one sperm that still has its intact acrosome can fertilize that egg. So at minimum, you would need about 11 sperm to get there, and maybe up to around 101 sperm to get there, just so that the one sperm can actually get in there and fertilize, while all the other ones help that one sperm to get in there and fertilize that egg. Okay. So the more you start with, obviously the more likely you're gonna end up with the right number at the right fallopian tube to fertilize that egg. Okay, now when you look at these secretions, you've got most of them coming from the seminal glands. I'm not gonna ever ask you to tell me what percentage, but most come from the seminals, then the prostate, then a little bit from the epididymis itself, and a little bit from those bulbourethral glands. Okay. Now, a couple other chemicals that you find in these secretions. Okay, you find some enzymes in these secretions. One of them is called, or there's a group of them called proteases. Proteases are helping to break down the proteins that are in that really thick mucus that I should have up here that are in the cervix. Have some in the vagina too, but also in the cervix. So these proteases, these enzymes, help to break down the proteins that help to make that mucus thinner so that the sperm can get through there easier. And then also you have enzymes in these secretions called antibiotic prostatic enzymes. They essentially are enzymes that help to kill bacteria, the bacteria that are in the vagina. Now again, sometimes females are real sensitive to their male partner's semen. And by killing these bacteria in the vagina can potentially cause other problems for other pathogens to grow there. Okay. Let me just give a sense of time. Okay, so then in terms of the sexual response, we're going to keep this real basic just because of time. So basically you can divide the sexual response into two key parts. You've got the erection phase and the ejaculation phase. The ejaculation we've already talked about, the key events. That's what I wrote up here. So again, this is propulsion of semen through and out the urethra. And that's going to be under sympathetic control. Smooth muscle sets up peristaltic contractions. Smooth muscle is going to cause the bladder sphincter to contract and glands to release their secretions. Okay. Let's say a couple things about the erection phase. The erection phase is all about engorgement of blood into that erectile tissue. That's what the erection phase is mostly about. And here we're not going to get into the details of it, but what I do want you to know about this phase is this is under parasympathetic control. Okay, this is a parasympathetic reflex. And we won't get into any more details about it. But I want you to kind of think about that for a second. So if your parasympathetic division is typically your rest and digest division, and your sympathetic is your fight or flight, typically speaking. Let's say you have a male that's very stressed out, right? There's a lot going on. He's stressed about this, that, and the other, and he's in sympathetic mode way more than he should be. If that's the case, he may have a difficulty getting an erection because there you need, at least for a while, you need that sympathetic division to be less active and your parasympathetic more active so you can get that parasympathetic reflex triggering vasodilation and filling that erectile tissue with blood. If the sympathetic division is so strong and we can't get the parasympathetic going, then again, erections may not be possible at that point. Okay. All right. Now, we're going to then look at the life cycle of humans. And again, we're going to keep this real basic. I'm going to say a couple things about mitosis and meiosis, keeping again that real basic. And then we're going to talk about how you make sperm cells. Now, making sperm and making eggs that we'll talk about tomorrow, that process is called gametogenesis, making of gametes. And you're going to see questions about gametogenesis on your lecture exam and on the practical. And as we're going through gametogenesis or spermatogenesis, I'll give you some examples of questions that you would see on your practical. Okay? And when we're up in lab, I'll double check and see how many questions from spermatogenesis. I think gametogenesis in general, I think it's 10 questions, but I'll double check when we're upstairs. Okay, so before we get to that though, if you look at the human life cycle, okay, let's start with fertilization. Okay, so for fertilization, essentially what you're going to have is you're going to have your gametes. You're going to have dad's sperm and mom's egg come together. Okay, in particular, the sperm is going to fertilize that egg, and as part of that process, what you're going to get are the chromosomes of each of those cells mixing together. So when you look at the sperm, there are 23 chromosomes in that sperm cell. That is an entire set of chromosomes. Okay? So that is one set of chromosomes. And a single set of chromosomes is referred to as N. Okay? And for humans, N is 23. 23 chromosomes is a set of chromosomes for humans. So if you look inside that sperm cell in its nucleus, you will see one chromosome number 1, one chromosome number 2, one chromosome number 3, one chromosome number 4, all the way down to one chromosome number 23, okay? One set in there. Those kinds of cells, when you have a set or N, are called haploid cells. Haploid just means they have a set of chromosomes. If you look at the egg, okay, and here I'm going to call the egg the ovum, and again I'll clarify all that stuff on Wednesday. The egg also is a haploid cell. It also has 23 chromosomes. It has one set of chromosomes. It has N chromosomes. It is a haploid cell. So when that sperm cell fertilizes that egg, as part of that process of fertilization, the chromosomes mix. And when you have these chromosomes mixing, you end up with the first cell of a new organism. And that first cell of a new organism is called the zygote. So the zygote is the first cell of the offspring. And that cell has 2 N chromosomes, right? It has 2 times 23. Or 2N is referred to as pairs of chromosomes. Or you could say 2 sets of chromosomes, okay, either way. Cells that are 2N are referred to as being diploid cells. So if you look in that zygote, there will be 2 chromosome number 1s, 2 chromosome number 2s, 2 chromosome number 3s, all the way down to 2 chromosome number 23s. Okay. So one of those sets of 23 came from dad, one of those sets of 23 came from mom. Now you've got the first cell of the new organism, that is diploid. Now what happens is this zygote, okay, this diploid zygote will start undergoing mitosis. And what we'll get is, instead of having one zygote, we will now end up with 2 daughter cells. And each of those daughter cells will be diploid. They will have exactly the same 46 chromosomes as that zygote had. And then each of those cells will undergo mitosis, and those resultant daughter cells will have exactly the same 46 chromosomes as their parent cell did, as that original zygote did. And each of these daughter cells will undergo mitosis, and you just keep doing that until eventually you end up with a multicellular diploid adult. So that means if I take any cell in your body, as long as those cells aren't sperm or eggs, as long as they're not gametes, as long as they're not sex cells, if I take any 2 cells in your body, okay, and cells that aren't sex cells are called somatic cells. So you have somatic and sex cells. Sex cells, again, are gametes. These are going to be in your gonads, testes, or ovaries. These are sperm and the egg, which is technically the ovum. If I take any 2 cells in your body that aren't these sex cells, then those are somatic cells. So let's say, for example, I take a muscle cell and a neuron. Okay, any 2 cells, it doesn't matter. As long as they have a nucleus. And and I look in their nuclei, okay, I look in each of their nuclei, what I will see are 46 chromosomes. I will see 46 chromosomes in that muscle cell's nucleus. I will see 46 chromosomes in that neuron's nucleus. And they will be identical to the 46 chromosomes from that original cell of that organism, from the zygote. So basically, mitosis just makes clones, okay? Mitosis is making clonal offspring. All right, now, to get gametes, the way that you get gametes is through a type of cell division called meiosis, or meiosis. For humans, the only time we do meiosis is to make gametes, that's it. We don't do meiosis any other time. If we need to do cell division because we've got cell damage, because we are growing, we do mitosis, okay? The only time we do this other kind of cell division, meiosis, is to make gametes. Now, to make gametes, what we'll do is we'll take a diploid cell, and from it, by doing meiosis, we will end up with haploid cells. Okay, and we're going to walk through that. We're not going to talk about the details of it. Hopefully, at some point, you learned it in bio, so you have some understanding of it. We're not going to hash out all those details again. Okay. All right, so what we're going to do is look at how you make spermatozoa. If you want to go back and look at mitosis and meiosis again, here's a reference slide with images of what's happening to your chromosomes. But this is purely a reference. There's nothing from here I'm going to come back and grill you on. Okay, that was bio. Okay, we're doing that all over again. Okay, so what we're going to do is we're going to walk through spermatogenesis. Okay, we're going to make sperm cells. Now, I'm going to write a lot on the board for this. And when we go to do this for females on Wednesday, we're going to follow the same basic template with a few key differences. But it's going to be the same basic idea. Okay, so first of all, what I'm going to put up here is to make sperm. I'm going to use this board for meiosis and mitosis. That's going to be this board. Over here, what I'm going to put is production. And again, I'll explain what that is in a general sense, and then we'll talk about some details of it. And then lastly, we do something called spermiation. This whole process takes 9 weeks. So about 9 weeks. For males, this is going to start at puberty, and then it's going to continue basically throughout the life of the male, as long as FSH, LH, and testosterone stay in normal homeostatic range. And we'll talk about how that happens at the end of today's lecture. Okay, so in a general sense, just take a drink here. Okay, in a general sense, what we're going to do with spermatogenesis is this is going to be where we're going to take what's called one spermatogonia. We'll talk about this in just a minute. One spermatogonia, or gonium for singular. It's going to undergo cell division. It's going to undergo mitosis. And then we're going to have meiosis. And by the time that we're done with this division, which we're going to draw out over here in just a minute, by the time that we're done, we're going to have 4 what are called spermatids. For every 1 spermatogonia, we're going to end up with 4 spermatids. And the whole point of spermatogenesis is is all about cell division, okay? And it's, I'm going to say the function, let's say the goal. Okay? The goal here is to get 23 chromosomes in each final cell. And we want to get 23, I'm going to put this down here, unique chromosomes. Actually, I'm going to put unique alleles. That's what I'm going to put. And I'll put that parentheses and then explain that. So what I mean by that is, if you look at mitosis, I said that mitosis is basically just cloning. You're taking 1 parent cell, and from it you make 2 daughter cells that look identical to one another, and they're identical to the cell they came from, okay? And then each of those daughter cells will undergo mitosis. You just keep doing that over and over and over. But all the chromosomes will be exactly identical from the cell that started the whole process. For meiosis, what you want to do is you want to end up with every gamete with 23 chromosomes. But you want the alleles in those 23 chromosomes to be different from one another so that your brothers and sisters look similar to you but different. So for example, if I look at the gene for hair color in 4, let's say, sperm cells, and depending on what the parent was, obviously, but potentially I might have, in 1 sperm cell, I might have an allele for blonde hair. And in another sperm cell, if I look at the gene for hair color, there might be allele for brown hair. And in another sperm cell, there might be, the gene for hair color might be a black hair allele, something like that. Again, it depends on what the original parent was, but you want those alleles to be different from one another. You want them all to have 23 chromosomes, but you want what they code for to be different. Okay? So that your brothers and sisters, again, are similar but different from you. That's meiosis. That meiosis is going to give us that. So spermatogenesis is all about divvying up chromosomes, getting chromosomes where we want them to be. That's all about spermatogenesis. What's in the nucleus? That's what we're doing here. Okay. Spermiogenesis is going to be where we take a spermatid, and what we're going to see when we get done with spermatogenesis is we do not have a spermatozoa yet. What we have is just a round cell called a spermatid. What's great about the spermatid is we've got those 23 chromosomes in the nucleus the way that we want them. That's what we've done in spermatogenesis. But our cell is just a round cell. It's a big old round heavy cell. It's way too big, doesn't have a flagella, doesn't have a head on it. It is not a spermatozoa yet. What we're going to do in spermiogenesis is we're going to take that spermatid and we're going to change the shape of that cell. We're not doing any cell division. Our nucleus is exactly how we want it to be. But what we're going to do is in our spermatid with spermiogenesis is we are going to lose most of the organelles and cytoplasm. We're going to lose most of the organelles in this cell. We're going to lose most of the cytoplasm in this cell. And what we're going to gain a head, a midpiece, and a flagella. And we'll look at an image of that. So that by the time we're done, what we're going to have for every spermatid that undergoes spermiogenesis, we're going to have a spermatozoa, a sperm cell. And it's gonna look like you think a sperm cell looks. Okay? And then the very last thing that we have to do is spermiation is where the spermatozoas, the sperm cells, detach from the wall of the seminiferous tubule and start swimming through the lumen to the epididymis. So spermiation is the detachment from the wall. That's what spermiation is. So now the sperm can take off to the epididymis. So this process occurs in the wall of the seminiferous tubule that's in the testes. Right. So now what we're going to do is we're going to look at the cells that are involved in spermatogenesis. I'm going to give you a second to get caught up with writing. I'm going to take a drink of water, and if you have any questions, you can ask me before we start looking at these cells. All right, so first of all, what we're looking at here, we're looking at part of the wall of the seminiferous tubule. So if this here is the lumen of our seminiferous tubule, then I said that the wall here was the wall of the seminiferous tubule. And this part here would be the part out by the hallway. And then this part here would be this part here. Part closest to the lumen, okay? So the part here that would be the deepest part of the wall, the part out where the hallway is, that's called the basal lamina. And then this part here is just the part closest to the lumen. So what we're gonna see as these cells are undergoing cell division, every time they do a round of cell division, the daughter cells will be deeper in the wall, and I don't know if I should say deeper in the wall. They will be moving within the wall, getting closer and closer to the lumen. So we're gonna start with cells out there by the hallway, and then every time they do a round of cell division, they're getting closer and closer to this side of the wall. So basal lamina is out by the hallway. This part here would be the part closest to the lumen. Okay. So we're gonna start with cells here, and then we're eventually gonna move this way. When we're done with spermatogenesis, this is what we're gonna have right here, spermatids. And then we're gonna do spermiogenesis, which takes these and turns them into spermatozoas. And then the last step of spermiation is where they detach, and then they start making their way to the epididymis. So what we're gonna do is we're gonna draw this out on the board right here and talk about these cells. We're going to talk about, are they diploid or haploid? We're going to talk about where are we in this process of spermatogenesis? Are we beginning mitosis? Are we ending mitosis? Are we beginning meiosis? Are we ending meiosis? And again, we'll see a similar thing for females with, it's going to be a little bit more complex with females, but same basic idea. So for males, we're going to start with a diploid cell, and those diploid cells are called sperm stem cells. We'll give them an official name in just a minute. And they are going to be up by the basal lamina, so the part out by the hallway. Okay, the part of the wall furthest away from the lumen. Okay, that's where these guys are going to start. And again, we'll put the name in just a minute. And then these guys are gonna undergo mitosis. And when they undergo mitosis, that one parent cell, that one sperm stem cell, will give rise to two identical diploid daughter cells. Okay, and again, we'll give them names in just a second. And then I'm gonna run out of board space here. So instead of trying to squeeze everything in this part of the board space, I'm gonna come over here, okay? But I want you to keep in mind, every time we do a rounded division, the resulting offspring are getting closer and closer and closer to the lumen, okay? So this up here is the basal lamina. But then here, I can't write anymore, so I'm just gonna make these guys come up here. But this is not the basal lamina over here, okay? This is just down here. It's just here. Okay, so we're gonna take one of those diploid cells, and it's going to undergo the first round of meiosis, which is called meiosis 1. And when you're done with meiosis 1, what you get are two haploid cells. And then each of those haploid cells is going to undergo the second part of meiosis, which is called meiosis 2. And each of those haploid cells will give rise to two haploid cells. So that when we look at spermatogenesis, we're starting with one spermatogonia, and we're gonna end up with 4 spermatids. I'm gonna fill in the names in just a second. Okay, so this cell that starts the whole thing, this is a spermatogonium. Spermatogonia for plural, spermatogonium for singular, they are sperm stem cells. Basically what that means is those cells can divide and differentiate and give rise to something that is much more complex than what it is. Okay? It can produce more specialized cells. That's what sperm cells can do. Okay? Now, what we're gonna give rise to are identical diploid daughter cells. And these daughter cells are very uniquely named daughter cell A and daughter cell B. Now, I want you to think about this for a second. For males, this process starts at puberty and has to continue throughout the life of the male. So that means if during puberty we took all of our sperm stem cells and turned them into daughter cell A and daughter cell B's and made all of these offspring, daughter cell A and daughter cell B, if they all went through meiosis to yield these spermatids, we would run out of these guys. Okay? So we have to have some way of doing this constantly. We have to somehow keep a population of spermatogonias up there at the basal lamina throughout the life of the male. So the way that we're gonna do that, and this is gonna be very different for females, because females aren't continuously doing this. For females, we actually do this in utero before we're ever born. Okay, this happens before we ever come out of the womb. For males, this happens from puberty throughout their life. Right? So the way that we're gonna keep this going is all the cells that are produced from mitosis of a spermatogonia, half of them, daughter cell B, we'll say, are going to do this process of meiosis. And the other half that are produced, we'll call daughter cell A, they're gonna stay up here at the basal lamina, and they're gonna become the new spermatogonia for the next round of mitosis. So that means every time we have a sperm stem cell undergo mitosis, half of everything that's produced stays at the basal lamina for the next round of mitosis, and half of everything that produced will continue with cell division and do meiosis. Now, when we look at daughter cell B, it's right here. I want you to notice it moves a little bit further down in the wall here to start meiosis. Now, this cell right here, daughter cell B, and this cell right here are the same cell, okay? We're just going to give this cell an official name as it starts meiosis. This cell and this cell are exactly the same cell. Nothing's changed about the cell, except this right here is just a little bit further down, closer to the lumen, okay? And again, I ran out of space, so I put it up there. This daughter cell B, we're gonna give it an official name as it starts meiosis, and we're gonna call it a primary spermatocyte. So primary spermatocytes are diploid cells. They are daughter cell B. Primary spermatocytes will undergo meiosis 1 to yield 2 haploid daughter cells that are called secondary spermatocytes. Each of those secondary spermatocytes will immediately go into meiosis 2, and each of those haploid secondary spermatocytes, they will each yield 2 haploid spermatids. And that is spermatogenesis, okay? Spermatogenesis is this cell division right here. Now again, these are not spermatozoa yet because we have to do spermiogenesis. We have to change their shape. We're not going to touch the chromosomes. We've got exactly the 23 chromosomes that we want in each of these cells. So on your practical, I'm going to ask you questions like this. Okay? I'm going to say, in spermatogenesis, what cell begins meiosis 1? And you will tell me that cell is called a what? A primary spermatocyte. If I say in spermatogenesis, at the end of meiosis 1, do you have diploid or haploid cells? What will you tell me?
Diploid?
At the end of meiosis 1, do you have diploid or haploid cells?
Haploid.
Haploid. What are those cells called at the end of meiosis 1? Secondary spermatocytes. Okay, and that's how I'll ask you questions about this, about your gametogenesis section on your practical, just like that.
What are haploid cells? Like
Just they have one set of chromosomes.
okay, this one.
Diploids have pairs of chromosomes. Okay, and N for humans is 23. Okay, 23. Okay, all right. So then let's look at spermiogenesis. So in spermiogenesis, we took that spermatid, and then we got rid of most of the cytoplasm, most of the organelles. We then gained a head, a midpiece, and a flagella. Now, on your test, I'm absolutely gonna ask you about those 3 parts, and I want you to know what those 3 parts correlate with. So the head is where you have your 23 chromosomes. The head is where your 23 chromosomes are located. At the tip of that head, you have your acrosome at the tip of that head. And in that acrosome, at the tip of that head, it's like a little helmet. That's where you have hydrolytic enzymes, digestive enzymes. These hydrolytic, so hydrolytic just means to break apart, okay? To do hydrolysis reactions, break apart things. Those hydrolytic enzymes their job, one of their key jobs, is to break apart the barriers around an ovulated oocyte. So that one sperm with its intact acrosome that didn't open it up and release those enzymes can fertilize the egg. Okay? You've got 10 to 100 sperm that will open up their acrosome, release those digestive enzymes, break down the barriers around the egg so that one sperm that never released its digestive enzymes, that still has an intact acrosome, can get in there and fertilize that egg. The mid-piece is where we have mitochondria. It's one of the few organelles we keep, mitochondria. Mid-piece contains mitochondria to make ATP. The tail is the flagella that's powered by the ATP made by the mitochondria that allows that sperm to move, to swim. And again, it cannot use its tail, its flagella, to get the whole distance. It needs peristaltic contractions to help. Now, we have to do all this. We have to make this sperm cell as light as possible, as aerodynamic as possible, because it's got a long distance to go. That's why it can't take any energy reserves with it. It can't take glycogen. It's got to use the fructose that's in the semen. It's got to transport it internally and then use it in the cell. What this also means, though, is when you go to look at a sperm cell fertilizing an egg and the first cell of a new organism is the zygote, really all the sperm cell is contributing to that zygote are 23 chromosomes. It really has no cytoplasm to contribute. It really has no organelles to contribute to that zygote. It's really just giving 23 chromosomes, which are important. But what that then means is when we go to do this for females, we can't have one of these yield 4 of these. What we're going to have with females is one of these is going to yield one huge cell. One big cell that compensates for that thing being so small. The ovum has to be twice as big as a normal cell because that's so small. The ovum has to bring twice the cytoplasm, twice the organelles, plus the 23 chromosomes. So that when the two cells come together, you end up with a zygote that has 46 chromosomes, 23 from here, 23 from mom, and then enough cytoplasm and enough organelles to start dividing. And all of that's coming from the egg, the ovum. So we're gonna see how does this shift in females so that we don't end up with tons of eggs, but we end up with one great big gamete. Okay, we'll talk about that later. Okay, let's start kind of tying things up here. So what we need to talk about to wrap this up is we need to talk about these really weird cells called nurse cells, and then we need to look at the hormones that regulate this process. So we've got these weird cells. We've never seen anything like these. These cells are called nurse cells. They're also called sustentacular cells. They're also called Sertoli cells. They're very odd cells. You can have in the wall of the seminervous tubular, right? If in one ejaculate you have 500 million sperm, you've got all these cells to get to those 500 million, and that's happening constantly. You've got that many cells being produced. So if you just think about hormonal regulation, you could not have hormone receptors on every single one of these cells to try to regulate that process by putting receptors on all of these. You would need so much hormone and so many receptors, you'd need a whole nother system to manage that. So instead what you have is you have these nurse cells. They're the ones that have the receptors for the hormones. these cells are undergoing cell division in these nurse cells that are receiving the hormonal signals. These nurse cells are attached through tight junctions, and what that does is it keeps the lumen fluid that's high in, the fluid here, the lumen, that's high in androgens, estrogens, ions, amino acids. It keeps that very different from the fluid out there, the interstitial fluid. And again, it's because we have to have so much division taking place constantly. We need this environment here to be very different from the environment out there. And nurse cells are gonna help to make sure that that happens. Nurse cells also are acting as a barrier to the immune system because during early development, these cells didn't exist, right? We didn't have these cells. This starts at puberty. So in early development, we didn't have these cells. In early development, when the immune system is developing and doing its checks and balances, where it says, I'm a T cell that responds to antigen E. And then it checks it against your body to say, is there an antigen E in the body? And if it is, then those immune cells get destroyed so they don't kill your very own cells. If you don't have antigen E, then those immune cells get to stay and develop because that means that's a foreign antigen to you. But any self-antigens, those T and B cells that would have target your own antigens, they get destroyed early in development. But we can't do that with these guys because they were never around. So what that means is these cells could very well be expressing antigens that you have T and B cells that would target and destroy those. So to make sure that doesn't happen, these cells are dividing inside a cell where your white blood cells and your T cells and B cells can't see these cells. So your nurse cells protect them. They act as a barrier. We already said that these nurse cells are going to have receptors for FSH and testosterone that will then set up an environment to support mitosis and meiosis so those cells can start dividing. When those cells are getting rid of, when those spermatids are getting rid of all those organelles and all that cytoplasm. And remember, we're talking about billions and billions and billions of cells, their organelles and cytoplasm. Where's all that stuff gonna go? How's it gonna get broken down? We can't have white blood cells just sitting in the testes breaking all that stuff down. They would do nothing else. So instead, these nurse cells break it down. These nurse cells are big enough to incorporate all that extra cytoplasm and organelles, and they can either use it or break it down with their lysosomes. These nurse cells produce a hormone called inhibin, and we'll see how inhibin works in just a minute. It's gonna work as a redundant mechanism with testosterone to help maintain levels. We'll talk about that in a second. And then probably the most interesting is these nurse cells secrete a molecule called MIF. So let's think about this for a second. We don't have tons of time we don't have time to go into development, but if you ever have time to study and learn about development, particularly sexual development, it's really, really interesting. But early in embryonic and fetal development, there will be a point in time where regardless of what your sex chromosomes are, so typically, if your 23rd chromosomes are XX, then typically that has been viewed as being a female, right? And if your 23rd chromosomes are XY, then typically that's been viewed as representing male. Now, early in all of our development, whether you have XX or XY, there will be a period of time where there is tissue that could become female reproductive structures at the same time that there's tissue that could become male reproductive structures. They're both present, okay? It's tissue that can divide and differentiate and become female reproductive structures, and right beside it, tissue that could become male reproductive structures. And the default mechanism, if there's no special chemical being presented, then this male tissue will die off and the female tissue will form. Now, if, however, you are genetically XY, okay, so this is what you have, then that means that during your development, you will have nerve cells. XX doesn't have nerve cells. So early in fetal development, you'll start making these nerve cells. And these nerve cells will then make what's called MIF, Mullerian inhibiting factor. Okay? And it goes by something else more commonly now, and I'm blanking on what that is, but you can look it up pretty easily. So what this MIF does is this MIF comes over and it binds to receptors and it kills off all the tissue that would have become female reproductive structures. So what you're left with is the tissue that will then become male reproductive structures. So nerve cells are pretty important for a lot of reasons, right? So this being just one of many reasons why it's important. Okay, so lastly, we want to look at hormones, right? So we're going to talk about this stuff here. I'm going to use, I think, this as a reference slide. Let's see. Yeah, we'll say a couple things. Yeah, we'll say a couple things like this. Okay, I'm going to erase this and use this board to draw our hormonal axis up here. And then when we get upstairs, we're going to look at a slide of the spermatocord, the testes, and the epidermis. Okay, so what we've got, we've already gone over this when we talked about hormones, so we'll just review this. We've got this hormonal axis. So we've got the hypothalamus, we've got the anterior pituitary, and we have the testes. Now, we're going to divide the testes into seminiferous tubules with interstitial cells around it. Interstitial cells around the seminiferous tubules. And really, what we should be thinking is, in the seminiferous tubules, we have nerve cells. Nerve cells are in the wall of our seminiferous tubules. It's the nerve cells that are in the wall of the seminiferous tubules. Okay. And then we can put up here many cells of the body. Okay, so whether we're looking at males prior to puberty or males post-puberty, same basic idea, okay? What we've got is, we've got pulses every hour, hour and a half of gonadotropin-releasing hormone. Okay? That comes from the hypothalamus, travels through the hypophyseal portal system to the anterior pituitary. Okay? Gonadotropin releasing hormone. Every hour, hour and a half, boom, you get a pulse of it. Another hour, hour and a half, boom, another pulse of it. Hour, hour and a half, another pulse of it. Travels through the blood, goes to the anterior pituitary, tells the anterior pituitary to make and release gonadotropins. Two gonadotropins, FSH and LH. FSH will come to the seminiferous tubules, in particular to receptors in the nerve cells that are in the wall of the seminiferous tubule. That's where FSH goes. Follicle stimulating hormone. LH goes to the interstitial cells. Luteinizing hormone goes to the interstitial cells. Absolutely, I'm going to ask you multiple choice questions on your final on this. So LH from the anterior pituitary goes to the interstitial cells. LH tells the interstitial cells to make testosterone. Now, testosterone does multiple things, okay? Testosterone is going to go out to many cells of the body and be involved in things like red blood cell production. It's going to influence sex drive, sexual behavior, libido. Okay? It's going to influence, it's going to help to mature and maintain reproductive structures. It's going to help to, it helps to increase muscle mass. Many other things, right? Testosterone also is going to come here to the nurse cells in the wall of the seminiferous tubule. And when we have FSH and testosterone at the correct levels, that causes spermatogenesis to occur. All that cell division that we just talked about that's taking place in the nerve cells in the wall of the seminiferous tubule is under the influence of FSH and testosterone. Those two hormones must be present together, right? This is an example of permissiveness that we talked about before. If FSH is present but testosterone isn't, or isn't at the right levels, it won't happen. We won't make males won't make sperm cells. If you've got testosterone at the right levels but not FSH, one, unlikely, but if that were to happen, won't make sperm cells. If testosterone levels are too high or way too low, won't make sperm cells. The hormones have to be at the right levels together at the same time to make sperm. Okay, so you need FSH and testosterone to do spermatogenesis. Now, we also know that testosterone is going to inhibit the hypothalamus. Okay. So, we're going to talk about one other pathway, but we're going to say testosterone, so in males, testosterone regulates its own levels by working through a negative feedback pathway, or you could say loop. Okay, and we did this before. We did it with thyroid hormone, but we said testosterone works the same way. So we're going to say this: if testosterone levels start to get too low, if testosterone started to drop, that means the hypothalamus would be less inhibited. And if the hypothalamus is less inhibited, then that means GnRH levels will start to rise, which means that FSH and LH levels will start to rise. And that rise in LH will cause the interstitial cells to make more testosterone, and we should be back to normal range. The opposite is true. Okay? If testosterone levels started to get too high, then the hypothalamus would be more inhibited. If the hypothalamus is more inhibited, then GnRH levels start to drop. If GnRH levels start to drop, then FSH and LH levels start to drop. And that drop in LH should cause the interstitial cells to make less testosterone. And that should bring testosterone back to normal range. Now, I'm going to say one more thing about a hormone that I mentioned before that comes from the nerve cells called inhibin. I'm going to mention that. But before I do, I just want to say that this axis, this hormonal axis, is in play prior to puberty. But prior to puberty, what we see is testosterone levels are too low to trigger spermatogenesis. That's why prior to puberty, boys aren't making sperm cells. They might have secretions, but there aren't sperm in there. Because testosterone levels, even though they're still being produced, are too low. Now, how does that work? If testosterone regulates its own levels, how do we keep testosterone low prior to puberty, but from puberty to adulthood, it's high enough that we can do spermatogenesis? And the way that works is the same pathway. It's just that prior to puberty, so childhood, right? And this really is from during in utero through childhood, but we'll focus just on childhood. So in childhood, the hypothalamus is strongly inhibited by testosterone. The hypothalamus is always inhibited by testosterone, but prior to puberty, it's strongly inhibited. So that means prior to puberty, during childhood, that keeps testosterone levels low. Not zero, we need testosterone for normal development, childhood development. But it keeps the levels low enough that spermatogenesis is not triggered. And then what happens is during puberty, the hypothalamus becomes more, no, I'll say it this way, becomes less sensitive to testosterone. Still inhibited by it, just not as strongly inhibited by it. It probably has to do with receptor numbers. There's probably less receptor numbers once you start hitting puberty. And so what this does is it allows testosterone levels to rise. It allows testosterone to rise and get and obtain, let's say obtain a new set point. And that'll be maintained from puberty throughout the life of the male. So prior to puberty, the set point is just more sensitive. It's more sensitive to testosterone. From puberty throughout the life of the male, there's a new set point that still, the hypothalamus is still inhibited by it. It's just not quite as sensitive to it. It's not as strongly inhibited by it. And that will often take a couple years to get that new set point, but that's just part of puberty. That's one of the many things that happen in puberty, is we get this new set point at the hypothalamus where new levels will be adjusted based on how sensitive the hypothalamus is to testosterone. Okay, lastly, this is a pretty important pathway, right? Because if testosterone levels aren't right on the money within a normal homeostatic range, and FSH right in normal homeostatic range, then we're not making sperm cells. And for males, we need to be making sperm cells essentially all the time. So we've got a redundant, a backup mechanism, a secondary mechanism that's also going to help to maintain levels. And that is coming out of the anterior pituitary, FSH, we said, targets the seminiferous tubule. And with testosterone in FSH, we said that drives spermatogenesis. But what FSH can do at the nerve cells at the seminiferous tubule all by itself is cause a hormone to be released called inhibin. And inhibin, you can imagine what it does. It inhibits. What inhibin does is inhibin will come up here and inhibit the anterior pituitary. It's just a backup mechanism. It works the same way. So let's just think about how it might work. So let's say, for example, let's say if, where do we want to start? Let's say if FSH levels started to get too high. Well, if FSH is getting too high, that means LH is also getting too high. So I'm going to put that in parentheses, okay? Because those two do the same. They're from the same place. If one goes up, the other goes up. If one goes down, the other goes down. FSH and LH are gonadotropins. They go up and down together. So if, let's say, FSH started to rise, LH would also start to rise. And as FSH started to rise, that means our levels of inhibin would start to go up. Now, when inhibin goes up, that's going to inhibit the anterior pituitary, so that's going to cause more inhibition at the anterior pituitary. And if the anterior pituitary is more inhibited, then that will result in less FSH and less LH. So inhibin is controlling the anterior pituitary hormones, which ultimately influences testosterone as well. Okay? So if LH is going up and FSH, more inhibin will be released. That will cause FSH and LH to go down. When LH is up, you get too much testosterone. When it comes back down, testosterone comes back down. So it just works with this pathway, again, to keep FSH, LH, and testosterone all bouncing within homeostatic range. So if one of those mechanisms doesn't work, the other one can carry the burden, okay? But they usually both work together. And then here, just to follow that idea up, if FSH got too low, that means LH will get too low, which means testosterone will get too low. And if FSH is getting too low, that means your levels of inhibin would get too low. And if inhibin started getting low, that would mean less inhibition at the anterior pituitary, which would cause your levels of FSH to go up. Your levels of LH would go up, and your levels of testosterone will go back up. Okay, questions on any of that stuff? Okay, so I'm going to shut this stuff down. I'll go open up the lab. I will get our slides up. It won't take very long to go over those slides. They're pretty easy. And I'll see you upstairs shortly.